The alpha helix is one of our two most common secondary structures that we see in proteins. Now, as with all of our secondary structures, the alpha helix is formed in order to maximize hydrogen bonding between our peptide bonds and also minimize steric crowding between our amino acids. Now, for the alpha helix, it accomplishes this using this tight kind of helical structure. So, as we form this helical structure, so we take the hydrogen bond donor of one of our peptide bonds, and we're going to be forming that hydrogen bond with a carbonyl directly above it. So these are not adjacent amino acids in terms of sequence, but as we're twisting around for any particular amino acid N, it's going to be hydrogen bonding with the amino acid directly above it, or the N+4 amino acid, right? So if this was the first amino acid down here, this would be the fifth amino acid up here, hydrogen bonding to it. And we see the same pattern repeated throughout our alpha helix, right? So each of our carbonyls is pointing down, and it's hydrogen bonding to the NH of our amide that's pointing up. Again, you can see that repeated over here, and it's always hydrogen bonding with the N+4 amino acid, 4 amino acids later in sequence. So it's just coiled around itself so it can form these repeating hydrogen bonds. And again, you can see that up here, continuing as we go on up our structure. So as we're forming these hydrogen bonds, we, of course, end up with this twisted structure with our specific spacing of our amino acids. And we end up with this 5.4 angstroms between loops of our helix. So an angstrom is 10 to the negative 10th of a meter. So it's a very small distance between our individual loops here. Of course, it's not that small on an atomic scale, but it's of course incredibly small compared to anything that we're used to handling ourselves. Now, our helix is going to be coiled in one specific direction. We refer to it as a right-handed helix. Now, what that right-handed is referring to is it's referring again to the direction of the coiling. So if you take your right hand and you point your thumb in the direction of the amino acid sequence, so that would be pointing from the N terminus towards the C terminus, and you curl your fingers around, your fingers are curling in the same way that the helix is looping, right? So here we have the right hand with the thumb pointing. Again, this would be from the N to the C terminus. And you can see how it's curling around in the same direction that the fingers are of that right hand. If you have a left-handed helix, so this would be curling in the opposite direction, you would be pointing your left hand up, following the N to the C terminus. And again, you're looping around in the opposite direction. So all of our alpha helices are normally right-handed. We do have some rare left-handed helices, but they look a little bit different than our typical alpha helix. So we have a specific direction that we coil. We have a specific spacing of our amino acids in order to form those hydrogen bonds between our carbonyls and our nitrogens. And so, of course, overall, we end up with a very regular structure for our alpha helix. You can see here some of the different structural representations of our alpha helix. So here's that atomistic model that we saw before. Over here on the right, we have the space filling model showing what this would actually look like. We look at kind of the full space that each of our atoms takes up in terms of the steric hindrance there. You can see we do have room for all of our atoms. And one of the ways that we're able to make sure that we have room for everything is by having all of our R groups, which are marked in purple, pointing to the outside of our helix, right? So notice it almost looks like a bunch of grapes sitting on the outside of our helix. None of them are kind of buried on the inside, where then, of course, if you have a large R group, it would be interfering with the helical structure. You can especially notice that here in our top-down view. So this is looking down the axis of our helix, and you can see all of these purple R groups are all pointing away from the inside of our helix there. So the R groups are on the outside. They're then going to be available for interacting with other molecules, while also avoiding any of that steric hindrance that you would have if you had big R groups kind of sitting inside of the helix. Over here on the right, you can see a representation of what it looks like in terms of our different amino acids and how close they are to each other on the axis of our helix. So notice how close our 1 and our 4th and our 1 and our 5th amino acids are as it curls around, which of course is important because the 1 and the 5th would be the N and the N+4 that are hydrogen bonding to each other. So the first amino acid is going to be sitting close to the 5th amino acid, which is also going to be sitting kind of just above the 8th amino acid, and so on and so forth down the helix, right? So the amino acids that you're next to in sequence aren't actually the amino acids that you're next to in terms of the 3D structure of this alpha helix. And that's something that's very important to keep in mind as we learn more about our alpha helices. So as we form this helix, we end up with an inner diameter of roughly 4 to 5 angstroms. So the inside of our helix is very small, and nothing can really go inside of our helix itself. Again, indicating the importance for having those R groups pointing out away from our helix. The outer diameter of our helix is much bigger with those side chains. That ends up being 10 to 12 angstroms. So we have no space on the inside, or at least a very small space that nothing would really be able to fit into. And then our R groups are on the outside, and they have space to move around. So again, going back to that top-down view, our amino acids, of course, line up nicely between N and N+4. And it also lines up between the N and the N+7 for 1 to 8, and then to 15, and so on as we move down the helix. So let's say we wanted to make a helix that had one side that was more hydrophobic and another side that was more hydrophilic or more charged. So in that case, we don't want our hydrophobic amino acids to be all next to each other in sequence. We want them to be next to each other on the same side of our helix, right? So we'd actually want to carefully place them such that they are all kind of N and then N+7 and so on, in order to get them kind of next to each other on the same side of our alpha helix, right? So if you actually wanted to build a helix with a hydrophobic side, you would want to do something like this. So this drawing over here on the left is what we call a helical wheel projection. And it's just showing all of our amino acids for a particular alpha helix and kind of where they are in terms of what side of our helix they're on as that helix is kind of spiraling down. So if you'll notice here, we have 1/2 of our helix that's all yellow. These are representing our more hydrophobic or nonpolar amino acids. And one side that's kind of green and red and blue. So these are either charged or neutral but polar amino acids to create a very polar side of our alpha helix, right? So it's a hydrophobic side and the hydrophilic side. And again, if you look at the numbers here, so you'll notice that the hydrophobic amino acids are not next to each other generally in sequence. We do have a couple that are next to each other, such as this alanine and tryptophan, but mostly they're kind of spaced out such that they all end up kind of being in a line on that same side of the helix. So again, if we go from 2 to 2+7, that would be the methionine here, amino acid 9, which is just what we want in order to get those hydrophobic amino acids near each other on the same side of our helix. And you'll see that repeated for other amino acids here as well, right? They're tending to be lined up kind of just with the the N+7 amino acid to put those hydrophobic amino acids on the same side. So if we look at this using a model, so this might look something like this, where we have our helical structure represented with this ribbon. This is one of our common ways to represent alpha helices. We have one side painted red, that's where all those hydrophobic amino acids are, and one side painted blue, where all of our hydrophilic amino acids are. This sort of helix, we would call this a amphipathic helix, which is a helix that has, again, both hydrophilic and hydrophobic portions. This would be able to then interact with something that's both hydrophobic on one side and then interact with something hydrophilic on the other side. And this sort of thing is something that we see quite often in different proteins, right? They'll have different regions of a helix that might have different properties in order to allow it to interact with different molecules. Now, another interesting property of our alpha helix is that we can have a helix dipole building up across our alpha helix. So if we look at our individual peptide bonds, so one thing you'll notice is that, of course, between our carbon and our oxygen, we have a dipole that's oriented towards our oxygen because it's more electronegative. Between our nitrogen and our hydrogen, we also have a dipole here oriented towards our nitrogen because it's more electronegative than our hydrogen. Both of these dipoles are pointed in the same direction, right? They're both pointed up in this case. Now remember, when we're forming our hydrogen bonds here, all of these are oriented in exactly the same direction, right? If you looked at that earlier figure that I showed, you would see that all of our hydrogen bonds here, right, they're all kind of pointing in the same way, right? It's always the nitrogen pointing up to the carbonyl that's pointing down, or vice versa, depending on your perspective. So because all of our peptide bonds are oriented in the same direction, and they all have a dipole that's oriented in the same direction, those individual bond dipoles all add up across the entire helix. So you've got all of these little dipoles, right? So each of these dipoles pointing up along our helix. And these dipoles all combine together to create a large dipole for our helix overall. Right? Remember, that's one of the things that we see with dipoles in general, right? Our molecular dipole is from an addition of all the individual bond dipoles of our molecule. We've got the same thing here, except on a bit of a larger scale, right? This is a very large polypeptide, or a very large peptide, and we have multiple individual bond dipoles all adding together and creating this large dipole for our helix overall. So we end up with one end of our helix that's going to be more negatively charged. We'll have a partial negative charge from that dipole, and then one end that's more of a partial positive charge. And this will be aligned according to the N to C terminus direction that we typically use with our proteins. The N terminus N will be the more partial positive end, and the C terminus N will be the more partial negative end. These sort of helix dipoles can have important roles in some of our different proteins. So we can use this dipole to help stabilize different charged molecules that we might bond to with our protein. Some of them are also used to help sense the charge state within the cell. So it's a very useful property of our helix that our proteins can take advantage of. Now, so far, with everything that we've looked at, we haven't really looked at the side chain at all, except to say that the side chain, right, is pointing out away from the helix, right? So for the most part, our amino acids don't directly affect our alpha helix, right? You could have any amino acid, and its side chain is just sticking off the side of our helix. It doesn't really affect the helix structure itself. So in general, sequence doesn't have a huge effect on our helix stability, but there are a few key exceptions. So a few of our amino acids can be considered helix breakers because they disrupt the normal structure of our alpha helix. So one of our helix breakers is proline. So proline, remember, has this strange ring structure where the side chain actually connects around back to the nitrogen of our peptide bond, right? So we have that alpha carbon, and we have it bonded out around, and it comes back to our nitrogen. And then our nitrogen, of course, would be bonded to our carbonyl carbon as normal. And we would head on down our amino acid sequence. Now, the problem here is that because we have this extra bond here to my side chain, right? My side chain, the R would be this little ring here. We don't have that hydrogen to act as a hydrogen bond donor. So we have no hydrogen bond donor. And so because of that, we end up missing one of our hydrogen bonds in our alpha helix, right? There's no hydrogen bond here between this amino acid and the 4 amino acids above it. So that missing hydrogen bond will affect our structure. We also have an extra constraint for our phi and psi angles because of this extra bond coming in here, right? We have this ring structure that's going to be somewhat limited in shape compared to your standard amino acid. So there's two separate constraints there that make it so our proline really doesn't like to be in alpha helices, right? It tends to destabilize our alpha helix. Again, our two key properties that we think of when we look at secondary structure: does it form hydrogen bonds, and does it avoid steric hindrance? Our proline hurts both of those two key attributes, right? It reduces the amount of hydrogen bonds that form, and it also makes additional problems with our steric hindrance because it has limited phi and psi angles that it can take up with that ring structure there. So in general, we won't tend to find prolines in many alpha helices. Now, the next helix breaker is actually a little bit surprising. So glycine is actually also a helix breaker. Now, glycine, of course, doesn't have the same constraints that proline does. It still has a hydrogen attached to its nitrogen, so it can form hydrogen bonds just as before. So the problem with our glycine is actually that it's too flexible, unlike our proline that was a little bit too inflexible. So you might think this is a bit of a funny problem, right? Because flexibility should reduce that steric hindrance and make it easier to form our alpha helix. The problem is that since our glycine is so small, it's actually able to take essentially whatever psi and phi angles it wants. So if we look at a Ramachandran plot for just glycine instead of our typical Ramachandran plot, we end up with something like this. So you'll notice we have green areas representing angles that don't see steric hindrance all over our Ramachandran plot, right? Basically, all 4 of our corners all have areas where we won't see a lot of hindrance. We only have this white area in the middle where we'll still see some steric hindrance. Now remember, our alpha helix normally like to sit over here in the bottom left corner. Well, glycine is so flexible that it doesn't really need to bend into that specific alpha helical shape, right? It's perfectly happy to sit over here or up here. And all of those, of course, aren't our alpha helical structures, right? So our glycine just tends to rearrange itself and try all sorts of different conformations, since it doesn't really hurt it to go into these different conformations, since it doesn't have any, or it doesn't have as much steric hindrance at any rate. So our glycine and our proline are both considered helix breakers, but in fact for almost opposite reasons. So glycine and proline are both considered helix breakers that prevent our amino acids from taking up an alpha helical structure. While these two amino acids are the primary ones that affect our helical structure, we do see that other amino acids will have a small influence on whether or not we take up that helical structure. In this table here, we see our various amino acids, and we have a delta delta G, which is representing the change in free energy of that amino acid if it was going to occupy an alpha helix compared to alanine. So alanine is used as our reference point, our standard here. Alanine is the best out of all of our amino acids for forming that alpha helix, right? It's still relatively small with just that little methyl group as its side chain, but it's not too small like our glycine. So alanine is the best at forming alpha helices, but we can actually look at these measured values for each of our amino acids and see how likely they are to form alpha helices. Of course, right away, glycine and proline, as we've already talked about, you can see they don't like to form alpha helices at all. We have a subset of various polar as well as some non-polar amino acids that also don't particularly like to form alpha helices. So some of these are quite large, that can influence the structure. Other of these have hydrogen bonding and stuff that can influence that structure as well. We'll still see them in alpha helices, but they're slightly less likely. Now, we also have some charged amino acids listed on here. So we have lysine, arginine, glutamate, and our here we have histidine down here as well. Aspartate is not shown on this chart. So you'll notice that the delta delta G here varies between these different amino acids. Again, that is partially based on the different sizes, right? The size can affect the likelihood to form that helix. But there is another factor that we need to consider for charged amino acids. And this is what the local charge density is, right? So if we have a single lysine, we can see with this 0.63 here that lysine is fine occupying an alpha helix. But if we had a bunch of lysines next to each other, these would all be positively charged, and they would all repel each other, right? So if we have very high like charge density within a specific region, that region is less likely to take a tight alpha helical structure, or even some of our other beta, or other secondary structures that we'll talk about later, such as the beta sheet. And instead, it's more likely to be in a more free random coil conformation, where those positive charges can kind of try to separate as much as they can from each other. So even though we have low numbers for some of these charged residues, in general, if we have very high charge density, that's also not going to prefer our alpha helical structure. So using this chart, we can't quite predict exactly whether or not a sequence is going to form an alpha helix. But it does show us some of the key things that we need to take into account when we're thinking about, is a sequence likely to form an alpha helix or not? At this point, it's actually pretty hard for us to predict exactly whether a sequence is going to form a specific secondary structure. So it's actually quite complicated just with all the different kinds of amino acids and then the different possibilities of how they can be arranged. It's really hard to actually calculate that. But again, there are some key things that we can look for, right? So proline or glycine, those of course don't usually form alpha helices. High charge density, that also is less likely to form alpha helices as well.